Today our lesson is within 2.4, Therapeutic Cloning, GCSE. Okay, before we talk about therapeutic cloning, let's just recap on what we said about stem cells. A stem cell is an undifferentiated cell, but it's more than that. Remember, there are lots of undifferentiated cells in your body, including things like the cheek cell. The difference with a stem cell is that a stem cell is undifferentiated but is able to differentiate and turn into any type of cell in the body. So of course, an obvious place to get stem cells from would be the fertilized egg. The fertilized egg has all the codes, all the genetic information, and they're all switched on. That one cell turns into 2 cells, turns into 4, turns into 8, 16, 32, and so on, until we have a small ball of cells called the embryo. At that point then, when we've got this embryo, this ball of cells, these cells are able to turn into all of the body cells, given the right conditions, because all of their genetic information in their genes is switched on. If we think about this, it's actually really quite special, because this embryo doesn't just go on to differentiate into your skin cell, your liver cell, your cardiac muscle cell, your heart cell, and those cells. What it does is it turns into the umbilical cord and the placenta. But over time, and a very short space of time, bits of the code that's contained in each of these cells gets turned off. And what that means is that the cell has become specialized. Specialized is the opposite of undifferentiated. How does it become specialized? Well, it produces proteins that are particular to that particular cell. It might change its shape and it will change its function. So its subcellular structures, the structures that are inside its cell, become different from a skin cell to a liver cell to a skeletal muscle cell. The other type of stem cell that you need to know about is the ones that come from bone marrow. Now, in adults, we can extract stem cells from our bone marrow. That's the center bit of your long bones. And in this region, you will also find stem cells in an adult. Now, when we talk about stem cells, they don't have the same ability as the ones from the embryo to differentiate into different forms of cells. They have limited capacity to turn into the full spectrum of cells, but they are able to turn into some different types of cell. And the added benefit of using stem cells from your own bone marrow is that you're not going to face rejection issues. So let's go back to the original idea of therapeutic cloning. Therapeutic means to fix something or make something better. And so what we're looking at is cloning to make something better. In other words, using bone marrow stem cells, injecting them in the body to fix things like paralysis, to fix diabetes, and to help people after they've had perhaps a heart attack. This is a new field in science, and they're experimenting all the time. Let's have a look at what they actually do. So if you get your stem cell, and if you get an embryo cell, here is the nucleus. What you're going to do is you're going to remove that nucleus from your embryo. So you're going to kick that nucleus out, so you haven't got one, okay? And what you're going to do is you're going to take your nucleus from your stem cell and you're going to move that into the empty embryo. What you're then going to do is you're going to take your embryo, put it in the right concentration of nutrients at the right temperature, and one cell is going to become 2, 4, 8, 16, 32, 64, 128, and before you know it, you'll have an embryo. Now, if we separate that embryo out into individual cells, each of them will have all the genes. They're all now embryonic stem cells, but they've been made from your bone marrow. So you won't face any rejection issues because it's got all of your genes in there. So if you then take this one and grow some cardiac cells, they're going to be genetically identical to your own cardiac cells. The same with this, you might grow a nerve cell, you might grow a cell from a pancreas, so you can inject that into your pancreas and replace your worn out cells that would normally make insulin but aren't. Let's just talk quickly about plant stem cells, which are called meristems. They are different from human stem cells. Human stem cells exist in our early stage of an embryo, or they persist in the bone marrow, and through the rest of your life they've gone. In plants, they keep their meristem, they keep their stem cells throughout their whole life, but they only actually occur in specific regions. In plant cells, you'll always find them in the growing root tip or the growing shoot tip, okay? So if you think about a plant, here is its leaves, and it's got a little growy bit there. Here is the soil. So this region here will contain stem cells, because this is the region that when the plant is growing, it's this region that grows. So that is part of the meristem, and just like on a shoot, what you're going to find is the top, the apex, this kind of region is where we're going to get the meristem. You can find it on fruit and vegetables too. So if you think about a carrot for a moment, we're all familiar with a carrot. They have that inside bit, that extra juicy bit. All of this is the meristem. All of this is the stem cell of the plant. And indeed, if you take a teeny piece of this and you put it in an agar dish and you put the right hormones and nutrients on it, you can just grow a leaf straight from it. Because that is the meristem, it's a stem cell, and from it you can form any type of cell. And it's the same with a cauliflower flora. You get these lumps of lumpy bits, the white bit of your cauliflower. If you take a piece of that, put it in a petri dish, put the right chemicals, the right hormones on it, you can grow any part of that plant, because it is the meristem, it's the stem cell of the plant. Now, why is this important? Who gives a hoot? Who cares? Well, lots and lots of drugs have been found from plants. So if you think about aspirin, aspirin was first discovered from the bark of the willow tree. So obviously we don't go to the bark of the willow tree now. Scientists have analysed the chemical structure and they're now able to make that in the laboratory, but it was discovered in the willow tree. Now we want to maintain our biodiversity because a lot of the compounds that we don't even know exist or can exist are found in plants and animals around the world. So when we've got rare species and they're in danger of dying out, or maybe you only find one of them, what you can do is you can take the meristem from that and you can then separate it out and grow up lots and lots of that type of plant. It's also of use in horticulture. Horticulture is the commercial growing of plants. Imagine you've got the best carrot ever. It's the biggest, it's the most orange, it's got lovely skin, it's not scarred by those horrible little lines, it doesn't grow any of those roots, you can just harvest it, and it's the most tasty carrot ever. Well, then what you can do is you take the meristem, again, you split that up. You can split it up into tens of thousands of plants and then put the right chemicals on, and they're going to grow into the genetically identical product. So in terms of horticulture, in terms of economic importance, then meristems and stem cells in plants are really, really important. Of course, later on what you're going to find is that by doing this, we reduce diversity, and that's not good, because should a new disease present itself, then the plants have got less genetic diversity to be able to combat that disease, and you can end up with a whole crop wiped out and not able to replace it. But you'll learn about that in later chapters. Okay, so you're doing AQA. AQA don't want to know what one thing is or another. They want you to be able to compare and contrast between them. What is a stem cell? You can't simply say it's an undifferentiated cell. You need to say it's an undifferentiated cell that can turn into any type of body cell. What's the benefit of an embryonic stem cell over a bone marrow stem cell? You need to know that. How can we use stem cells to treat paralysis, and how can we use stem cells to treat diabetes? How can rare species of plant be cloned to save them from extinction? And how can we use how can we use plant stem cells, in other words, meristems, to our economic advantage?